Hello everyone, this is Latha Akula. In this video we are going to study about Meiosis-ll. So in the last video we have completed Meiosis-1. In the Meiosis-1 what we have studied? The homologous chromosomes, they were separated and which will enter into the opposite poles and enter into the two different cells, right? So whatever the daughter cells created in the Meiosis-1, they starts the Meiosis-2. So those daughter cells only, those haploid daughter cells, this starts the meiosis-2, they'll act as the mother cell or the parent cell, right? How many daughter cells were formed in the meiosis-1? In the meiosis-1, we have two daughter cells. How many? Two daughter cells are formed, right? Those two daughter cells only, they starts the meiosis-2. Here we are doing this we are studying this division simultaneously for these two daughter cells. So in the meiosis-2 what will happen? This meiosis-2 is similar to the mitosis. Hence we can call it as meiosis-2 is nothing but mitosis or equational division because it is somewhat similar to the mitosis. But of course it is the stages will be similar. There are some dissimilarities also seen. If we see the mitosis occurs in diploid somatic cells or body cells. But whereas this meiosis-2 occurs in haploid germ cells or sex cells. Where the meiosis-2 occurs in the haploid germ cells or sex cells. Whereas mitosis occurs in diploid somatic cells or the body cells. Here the daughter cells formed are not similar to each other and they were not similar to the mother cell also, right? But whereas in the mitosis the daughter cells were similar to each other, they were identical to each other and they were similar to their mother cell also. These are some of the differences seen in the mitosis and meiosis-2. And here what is the main purpose of this meiosis-2? The separation of chromatids of the daughter cells of the meiosis-1 is the main purpose of the meiosis-2. So in the meiosis-1, homologous chromosomes, the paired homologous chromosome, they have been separated, which will be equally distributed into the daughter cells, right? But here in the meiosis-2, within the homologous chromosomes, the separation of chromatid separation takes place in the meiosis-2, and that separation that will be takes place in the two stages. One is the karyokinesis and cytokinesis, which is similar to the mitosis. But again in the karyokinesis also, how many stages are there? 4 stages. What are the 4 stages? Prophase 2, metaphase 2, anaphase 2, telophase 2, and which is followed by the cytokinesis 2. The first one is the prophase 2. Here prophase 2, if we see the prophase 1 of the meiosis-1, which is very large, long and elaborate with 5 stages, which is highly complicated, right? But whereas the prophase 2 is very short and simple. Okay? We cannot compare the prophase 1 with the prophase 2 because prophase 1 is much complicated and elaborate process, but whereas the prophase 2 is a short and simple process. What will happen here? The chromatids condenses and they will become compact. Here the nuclear membrane and nucleolus disappears. What will happen in the prophase 2? Nuclear membrane and nucleolus disappears and the chromatids will become compact structures, and the centrioles, they starts moving to the opposite ends. The duplicated centrioles, they starts moving to the opposite ends in the prophase 2. Now the cell enters into the, after completing the prophase 2, the cell enters into the metaphase 2. In the metaphase 2, what will happen? In the prophase 2, the nuclear membrane degenerated, right? Now there is no boundary for the chromosomes. Now the chromosomes that can freely move in the cytoplasm. Now the chromosomes, they align themselves at the equator region. The chromosomes align themselves, these two chromosomes align themselves in the equator region, and from the centrioles, aster formation takes place. From the aster, spindle fibers develop. These spindle fibers, they were attached to these chromosomes near the kinetochore region. Where the chromosomes have been attached to the spindle fiber? Near the kinetochore. Already we have studied about this in detail in the mitosis, right? That is why I'm not telling you in detail. So these chromosomes, they are attached to the, sorry, the spindle fibers attached to the chromosomes near the kinetochore region. So this is what happens in the metaphase 2. Now the cell enters into the anaphase 2. So not, we cannot call it as cells. Now the cells enters into the anaphase 2. So in the anaphase 2, what will happen? Here, these spindle fibers, they starts pulling the chromosomes. That means the length of these spindle fibers, decreases, and the spindle fibers, they starts pulling these chromosomes. Now what will happen? There is the splitting of centromere. Here in the chromosomes, what will be there? There is a centromere is present, right? The centromere starts splitting because of the pulling action of the spindle fibers. So in the anaphase 2, what will happen? Splitting of the centromere occurs, and after splitting, the chromatids, they move to the opposite ends. See here, the splitting takes place and chromatids, they were pulled to the opposite ends. This is about anaphase 2. Now the cell enters into the telophase 2. In the telophase 2, what will happen? The chromatids, they completely reaches to the opposite poles, and around these chromatids, the nuclear membrane and nucleolus starts reappearing, and the spindle fibers, they completely disappears. So what will happen in the telophase 2? Chromatids, they reaches to the poles, and the spindle fibers completely disappears. Nuclear membrane, nucleolus reappear, and finally 4 haploid nuclei are formed. How many? 4 haploid nuclei are formed, and these chromosomes, they starts uncoiling. That is, decondensation of the chromatids takes place in the telophase 2. For our convenience, we are drawing like the chromosomes. Actually, what will happen is decondensation or uncoiling of the chromosomes takes place here. Now, totally 4 nuclei are seen. How many nuclei? 4 nuclei are seen. And now the cell enters into the cytokinesis stage. Up to telophase 2, we have completed karyokinesis. That is, nuclear division is over. Now the cell enters into the cytokinesis 2. Cytokinesis 2 means what? Cytokinesis means that is separation of cytoplasm. Division of cytoplasm. If we have already studied in the animal cell, how the cell separates by means of furrow formation. So in animal cell, what we will see? By means of forming the furrow, the cells will separate. But whereas in the plant cells, there is a cell plate. By means of cell plate, the cells will be separated, right? Here, after separation, what will happen? Totally 4 haploid daughter cells are formed from the single diploid cell. From the single cell, we have already drawn in the last meiosis 1, right? So we have just seen the daughter cells. From this stage onwards, we are drawing here. So this stage already we have completed in the meiosis 1. So from one single diploid cell, how many daughter cells were formed? 4 haploid daughter cells were formed, and these 4 daughter cells in the meiosis 2, we can call them as gametes. Because they were participating in the sexual reproduction. If it is a male, we can call them as a male gametes. If it is a female, we can call them as a female gametes. Like this, we have completed the meiosis 2.